Hey everyone, thanks for tuning in to another deep dive. This time you guys sent us a really cool research paper. It's from Advanced Biology from 2025.
Oh, wow.
And the title is a mouthful.
Okay.
RhoA and Rac1 as mechanotransduction mediators in colorectal cancer. Yeah, I know.
I know.
I know it sounds like a lot,
Yeah.
but it's actually looking at something really, really interesting.
Okay. I'm intrigued.
Yeah, it's about the role of physical forces.
Physical forces.
Yes, physical forces
Okay.
and how colorectal cancer actually progresses.
Interesting. So not just like the typical genetic factors or chemical signals. We're talking about actual physical forces.
Exactly.
Okay.
It's this idea that it's not just about what's happening inside the cancer cells themselves,
Right.
but about like the actual physical environment,
Oh, wow.
the pressures,
Wow.
the textures they're experiencing.
Yeah.
And specifically, this paper is looking at two proteins, RhoA and Rac1.
Okay. RhoA and Rac1. Got it.
And basically, our mission in this deep dive is to like break this all down.
Yeah.
To help everyone understand, like, what are the key takeaways from this paper?
Absolutely.
How do these physical forces come into play? And what could this all mean for actually diagnosing this type of cancer?
That's a big one for sure.
Hmm.
Early diagnosis can make a huge difference.
Absolutely. So think of this like your shortcut to being really well informed.
Love that. A shortcut to knowledge.
Yeah, exactly.
I like it.
So we're stepping into this really cutting edge field
Okay.
called mechanobiology.
Like nanobiology?
Yeah, and it's all about understanding how these physical forces, these mechanical cues,
Yeah.
affect how cells behave.
Interesting. So instead of just thinking about cells reacting to chemical signals,
Right.
we're also considering how they respond to being, squeezed, stretched, pushed around, almost like they have a sense of touch.
Exactly, like they have a sense of touch.
Just why.
That's crazy, right?
It is.
So cells can actually sense, generate, and respond to these mechanical stimuli.
Wow.
And that has a huge impact on just like basic processes,
Yeah.
how they stick to each other,
Uh-huh.
how they move, how they multiply, and even what type of cell they ultimately become.
Right. That differentiation process. Fascinating.
Yeah, and when we're talking about cancer, all of these mechanobiological processes,
Yeah.
they're really deeply involved in how tumors grow,
Okay.
how they invade surrounding tissues, and then that really crucial part, how they metastasize.
Right. How they spread to other parts of the body.
Exactly.
And when we think about colorectal cancer specifically,
Yeah.
you can just imagine the kind of physical forces at play in the colon and rectum.
Oh, absolutely.
Right. I mean, you've got the constant movement of digestion, the natural expansion and contraction of the tissues.
It's like a really dynamic environment.
Exactly. And so when a tumor starts to grow there,
Right.
it not only disrupts that local tissue structure,
Uh-huh.
but it also significantly alters the mechanical environment.
Yeah, it's like adding another layer of complexity to an already complex system.
Absolutely.
Yeah.
And the idea behind this research is that these mechanical changes,
Yeah.
they can actually influence the signaling pathways within the cancer cells themselves,
Wow.
potentially driving the cancer forward, making it more aggressive.
So this is where RhoA and Rac1 really come into play.
Okay, so tell me more about those.
Yeah, they're described as crucial players
Yeah.
in this whole process. Small but mighty.
Small but mighty. I like that.
Yeah, so what are they doing?
Yeah, what's their role?
They belong to this class of proteins called small GT passes,
Okay.
and their main job is to regulate the cytoskeleton.
The cytoskeleton. So that's like the internal framework of the cell, right?
Exactly like the scaffolding.
Okay.
It's made up of these protein fibers like actin filaments and microtubules.
I've heard of those.
Yeah, and it gives the cell its shape.
Okay.
It helps move things around inside the cell,
Right.
and it's super crucial for the cell's ability to move.
Got it. So RhoA and Rac1 are like the conductors of this cellular orchestra of movement and structure.
I love that analogy. That's
Yeah.
perfect. So let's break down what each of them is doing specifically.
Yeah, let's get into the specifics.
Okay, so RhoA,
Yeah.
it's really key for cell adhesion and migration.
Okay.
It helps the cell form what are called stress fibers.
Stress fibers.
Yeah, these are bundles of actin that create this contractile force within the cell.
So they allow the cell to kind of pull on its surroundings.
Exactly, like little tiny muscles.
Cool.
I know, right? And then RhoA also plays a role in the formation of focal adhesions.
Focal adhesions. Those are like the anchors.
Yes, like anchors that connect the cell's internal skeleton
Yeah.
to the extracellular matrix.
The ECM, right? That network of proteins and molecules outside the cell.
Exactly. And these focal adhesions, they're not just for sticking.
Yeah.
They also act like sensors.
Wow. So the cell can actually feel the physical properties of its environment through these focal adhesions?
Yeah, it's amazing.
That's incredible.
I know. So it can feel like the stiffness, the tension around it.
So it's like the cell is getting tactile information
Yeah.
about its surroundings.
It really is. It blows my mind.
It's amazing.
Okay, so we've got RhoA kind of like holding on and feeling what's around it.
Right, the anchor and sensor.
What about RAC1?
What's that one up to?
So RAC1 is more focused on movement and invasion.
Okay.
It's involved in forming lamellipodia.
Lamellipodia, those are those sheet-like protrusions, right?
Yes, at the leading edge of a moving cell.
Okay, so it helps the cell to kind of crawl forward.
Exactly, like it's reaching out and pulling itself along.
Got it.
And RAC1 also helps create membrane ruffles.
Membrane ruffles now, those are interesting.
Yeah, they're these dynamic extensions of the cell membrane,
Okay,
and they help the cell move and navigate through tissues.
so it's all about mobility and exploration for RAC1.
Exactly. So RhoA is like grip and sense,
Uh-huh.
and RAC1 is push forward and explore.
I like that. A nice way to sum it up.
So you can imagine if these processes get messed up,
Yeah.
especially in a growing tumor, where the physical landscape is constantly changing.
Right, it seems like things could go really wrong.
Oh, absolutely. And that's exactly what the paper suggests,
Yeah.
that as tumors grow
Yeah.
and the mechanical environment of the colon changes,
Uh-huh.
the normal functioning of these RhoA and Rac1 pathways
Right.
can get disrupted.
Okay.
And this disruption could be a big factor in speeding up the progression of the cancer,
Oh, wow.
making it more aggressive and more likely to spread.
That's concerning.
Yeah, it is. And that leads us to the main question of the study.
Okay.
Should the levels of these proteins
Yeah.
in colorectal cancer samples,
Uh-huh.
especially when they're under mechanical strain,
Right.
could those levels actually serve as indicators or diagnostic markers for the disease?
So they're looking at whether the behavior of these proteins under physical stress could
Whoa.
tell us something about the cancer.
Exactly.
That's really clever, actually.
Yeah, so they're not just looking at what these proteins do,
Yeah.
but if their behavior can give us clues about how likely those cancer cells are to spread.
And if they can identify these proteins as diagnostic markers,
Right.
then that could lead to earlier detection and
Exactly.
hopefully better outcomes for patients.
Absolutely. And this is all super important because colorectal cancer,
Yeah.
even with all of the medical advances we've made,
Uh-huh.
it remains a huge problem.
It does. It's the third most common cancer globally,
Yeah, and the second leading cause of cancer-related deaths.
which is just a stark reminder of how much we still need to learn and how much more we need to do.
I know, and the study really emphasizes this concept of the tumor microenvironment.
Yes, the
Yeah.
tumor microenvironment.
This incredibly complex mix of not just the cancer cells themselves, but other types of cells.
Right.
The extracellular matrix that we talked about earlier.
The ECM.
And within this whole ecosystem,
Yeah.
mechanobiology is highlighted as this pivotal research area.
It makes sense. It's not just about the cancer cells in isolation. It's about how they interact with their surroundings.
Right? It's like this whole intricate web of interactions.
And those interactions are both biochemical and biophysical.
Yeah,
Yeah.
exactly. So the tumor microenvironment isn't just a passive backdrop.
Right. It's an active participant.
Exactly. It's shaping what's happening.
Absolutely. And mechanobiology is essential for understanding all of that.
So can you talk a bit more about how mechanobiology specifically plays out in colorectal cancer?
Sure. So as the tumor grows, right, it starts to physically compress the surrounding healthy tissues
That makes sense.
just by its sheer bulk. And at the same time,
Yeah.
the tumor cells themselves are often remodeling the ECM.
Uh-huh.
They lay down more of it in some areas,
Okay.
making it denser or stiffer.
So they're actually changing the physical properties of their surroundings.
They're actively shaping their environment. And then on top of that, you've
Right.
got the mechanical forces from digestion.
Right. The muscle contractions and all that.
Exactly. So all of these tumor-induced changes create this really altered mechanical landscape.
Yeah, it's like a constantly shifting terrain.
It is. And that altered landscape then feeds back onto the cancer cells,
Wow.
influencing their behavior through these mechanotransduction pathways.
It's like a vicious cycle.
It can be, yeah.
So RhoA and Rac1, they're really key components in this whole mechanotransduction machinery.
Absolutely. They're like the fundamental elements, the molecular switches
Yeah, so how do they fit into this larger process?
of cells sensing and responding to mechanical forces.
Exactly.
Okay, well, when a cell experiences a mechanical force,
Yeah.
like it's being stretched or compressed,
Uh-huh.
this physical stimulus can trigger a whole cascade of biochemical signals inside the cell.
Right.
And those signals can activate RhoA and Rac1.
Okay.
And once they're activated, they orchestrate changes in the cytoskeleton,
Wow.
allowing the cell to physically respond to that initial force.
So it's like a chain reaction.
It is, yeah,
Yeah.
a signal transduction cascade.
And the cell can respond in different ways.
Right, it might strengthen its grip, change its shape, or even start moving.
That's amazing.
It is. The cell is so dynamic.
And the cytoskeleton itself, it's more than just structural support, right?
Oh, absolutely. It's incredibly dynamic and adaptable.
Yeah.
It's constantly changing and reorganizing itself.
So it's made up of those actin filaments,
Uh-huh.
microtubules, and intermediate filaments.
Right, and together they maintain the cell's shape.
Yeah.
They transport molecules.
Like a highway system.
Exactly.
Yeah.
And of course, they enable the cell to move and interact with its environment.
It's mind-blowing how much is going on inside a single cell.
It really is.
So let's go back to RhoA and Rac1 and
Okay.
those specific functions.
Yeah.
So RhoA is associated with stress fibers
Uh-huh.
and focal adhesions.
Right.
How do those actually help a cell adhere to its surroundings?
Yeah, sense what's going on.
Well, those stress fibers,
Yeah.
those bundles of actin filaments,
Yeah.
they generate that contractile force within the cell,
Right.
allowing it to grip and pull.
Like tiny little hands.
Yeah, exactly. And then the focal adhesions, they're like the points where those stress fibers physically connect to the ECM.
Right, through those specialized proteins on the cell surface.
Exactly. And those connections aren't just passive.
No.
They act as sensors.
So the cell can feel the stiffness, the tension, all of that.
Yeah, it's incredible.
This is so cool.
I know. Okay, now Rac1, with its lamellicodia
Yeah.
and membrane ruffles,
Uh-huh.
how do those enable movement and invasion?
Especially in a cancer cell.
Well, those lamellipodia, remember, those are the sheet-like protrusions.
We're right at the leading edge.
Yeah, they allow the cell to extend its reach,
Okay.
kind of explore its environment.
Like putting out a foot to test the ground.
Exactly. And then those membrane ruffles, they also contribute to motility
Okay.
and help the cell navigate through those complex three-dimensional tissues.
So they're like little rudders.
Yeah. And in cancer, all of these RAC1-driven processes,
Uh-huh.
they're super crucial
Okay.
for those cells to break away from the primary tumor mass, to invade, and then eventually metastasize.
So they're really key players in that whole process of spreading.
Absolutely.
Now, the paper specifically mentions EMT,
Yes. Epithelial to mesenchymal transition.
invasion and metastasis.
These are like the hallmarks of cancer progression.
Right, the things that make it so dangerous.
But you explain EMT a little bit more?
Sure. So EMT is basically a process where epithelial cells, which normally form these nice tight sheets, they lose their adhesion and gain the ability to migrate and invade.
So they become more like mesenchymal cells.
Exactly. They're more mobile and less structured.
And this is a critical step for cancer cells to spread.
It is, yeah.
Yeah.
It allows them to break free and move to other locations.
And how are RhoA and Rac1 involved in this transformation?
Well, RhoA is really important for the cytoskeletal changes that happen during EMT. It helps to dismantle those tight junctions.
The ones that hold the epithelial cells together.
Exactly. And then Rac1, it fuels the invasive behavior. It provides that motility that those transformed cells need.
To move through the ECM and spread.
Right. So they're both working together, but in different ways to promote this transition.
And it's not just about what these proteins do on their own.
No.
The study also points out how mechanical stress within the tumor, whether it's from the growth changes in the ECM, or even forces from therapies, can actually directly affect things like cell signaling,
Makes sense.
gene expression, and the function of proteins.
So the physical forces are actually influencing the internal workings of the cells.
Exactly. It's all interconnected.
It's a complex system for sure.
So this leads us to the core of what the researchers were trying to do.
Okay.
They wanted to investigate how RhoA and Rac1 behave, specifically their expression levels,
Okay.
when they're subjected to mechanical strain in actual tissue samples.
From colorectal cancer patients.
Exactly. And they got those samples after surgery.
So they're using real clinical samples.
Yeah. Which is a huge strength of the study.
Absolutely. It's
You-
much more relevant than just using cell lines.
Right. They're looking at these proteins in a much more complex and realistic environment.
With all the different cell types,
Mhm.
the genetic mutations,
Yeah.
the actual tumor microenvironment.
It's the real deal.
It is.
So what did they find?
Well, the big takeaway
Yeah.
is that they found this clear overexpression of both RhoA and Rac1.
Okay.
And it was associated with the progression of the cancer.
So the more advanced the cancer, the higher the levels of these proteins.
Exactly. And they noted that this finding actually aligns with some earlier work.
Oh, interesting.
That was done on breast and liver cancer cell lines.
Okay.
But now they're seeing it in actual patient tissue from colon cancer.
So it's like adding more evidence to support this idea.
It is, yeah. It's really strengthening those findings.
Now, can you walk us through a little bit about how they actually did this experiment?
Sure. So they started by isolating the cancer cells from the tumor tissue that was removed during surgery.
Okay.
And then they placed these cells onto a special device made of PDMS, polydimethylsiloxane.
Okay.
It's a flexible, silicone-like material
Okay.
that's often used in biomechanical studies.
So it allows them to apply that controlled mechanical stretch
Exactly. They can mimic the forces that those cells might experience in the body.
as the tumor grows or tissues move.
Right. And they mentioned that the average cyclic strain was about 1.38%.
Okay.
So that's a measure of how much the cells were being stretched and released repeatedly.
Kind of like a gentle rhythmic flexing.
Exactly.
And to be sure, they were looking at cancer cells.
Yeah.
They used H&E staining.
Hematoxylin and eosin staining.
Right.
Which is a very standard technique in histology.
Yeah, you see it everywhere.
It is. It's a classic. So it allows you to visualize the different parts of a tissue sample under a microscope.
Okay.
And by looking at the morphology,
The shape.
the shape of the cells after staining, they could confirm that they were indeed dealing with cancer cells.
Based on those characteristic features.
Like a larger nucleus, prominent nucleoli, and irregular nuclear borders.
Got it. So they did that visual confirmation.
Yeah.
And then they also did some qualitative analysis.
Yeah.
Just by looking at the cells under a microscope.
Right after they had been stretched.
And what did they see?
They observed some interesting changes in how the cells were organized and connected after the stretching. They saw some cellular rearrangements and the formation of these distinct cell-cell connections.
Interesting.
And they mentioned that these observations were actually similar to what they had seen in previous studies with breast and liver cancer cell lines.
So this suggests that the stretching, the mechanical strain, can actually influence how these cells interact and organize.
Exactly. It's not just a passive response. They're actively changing their behavior.
That's pretty amazing.
It is.
So they also included some comparisons in their study.
Yeah.
They used a colon cancer cell line called LS513.
Right. A well-established one.
And a non-cancerous colon cell line called FHC.
Okay, so why did they include those?
Well, they provided control groups and reference points.
So they could compare how the primary cancer cells were behaving compared to both established cancer cells and normal cells.
Exactly, because those primary cells from the patient tissues, they're super valuable because they're more realistic.
Right. They have that heterogeneity, and they're influenced by the patient's own physiology, whereas cell lines are more uniform and easier to manipulate in the lab.
Right, so they each have their pros and cons.
They do.
So they also use fluorescent staining using DAPI and actin green.
Okay, so they could visualize the nuclei and the actin filaments?
Yeah.
Cool. What did they find?
Well, the different samples showed some interesting variations in their behavior.
Oh, okay. Tell me more.
So one sample, S1, it tended to form more tightly packed clusters after stretching.
Nice. Then S2 and S3.
Yeah.
They showed strong connections and clustering,
Okay.
but also this clear reorganization of those actin filaments within the cells.
Wow, so like active internal changes.
Yeah, and potentially more communication between the cells.
Interesting.
And then S1 and S5, they seemed to detach more readily after the stretching force was applied.
So different responses to the mechanical stress.
Yeah, and the study connected these behaviors to the stage of the cancer.
Oh, okay. So how did that break down?
Samples S1, S4, and S5.
Yeah.
Those were from patients with stage 2 colorectal cancer.
Okay.
And S2 and S3 were from patients with more advanced stages.
Got it. And what did they observe?
They found that the stage 2 cancer cells, they generally seem to have a more stable cytoskeleton.
Okay.
And they didn't exhibit as much reorganization in response to the stretching.
Interesting.
Whereas those advanced stage cells,
Yeah.
they showed considerable remodeling.
So more adaptable. Yeah. And the researchers thought this could be linked to their increased motility.
Makes sense.
And their ability to adapt to forces within the body.
Which could ultimately promote metastasis.
Exactly.
Now, they also quantified the percentage of cells that were lost after stretching.
Yes, they did.
Yeah.
What did those numbers look like?
Well, S1.
The one that tended to cluster.
Right.
It had the highest percentage of cell loss at 34%.
Okay.
And then the other primary cancer cell samples range from about 19 to 24% cell loss.
Got it.
The LS513 cell line showed a lower loss of 15%.
Okay.
And the non-cancerous FHC line had the lowest by far at just 4%.
Huh. So quite a range.
Yeah. And what did the researchers think about these differences?
Well, they suggested that the higher loss in the primary cancer cells might be because they still have those intact stress response pathways.
Including apoptosis.
Right. Programmed cell death.
So those pathways might be more easily triggered in the primary cells.
Exactly, when they experience that mechanical stress.
But those established cancer cell lines,
Yeah.
like LS513,
Uh-huh.
they often have mutations
Right.
that can disrupt those normal processes.
Making them more resistant to cell death.
Exactly. And the very low loss in the non-cancerous FHC cells was probably due to their more elastic cell membranes
Okay.
and their intact signaling mechanisms.
So they can cope with the stress better.
Exactly.
That makes sense.
Now let's talk about the actual measurement of the biomarkers themselves.
Okay. RhoA and Rac1.
So these proteins,
Yeah.
they can be found in different spots within the cell.
Right, depending on their state.
So when they're active and bound to GTP,
Uh-huh.
they're often at the plasma membrane,
Okay.
where they can interact with other proteins.
To reorganize the cytoskeleton.
Exactly. And when they're inactive,
Yeah.
bound to GDP,
Uh-huh.
they hang out in the cytoplasm,
Okay.
often with GDIs.
Guanine nucleotide dissociation inhibitors.
Right.
And they can also be found in the nucleus.
Yeah, where they can influence gene expression.
So they're busy little proteins.
They are. And to measure them,
Yeah.
the researchers had to break open the cells.
Relice them.
Exactly. And they used NP40.
A detergent.
Yeah.
To release the proteins.
Right. And then they used ELISA and flow cytometry
Ah, good choice.
to quantify those amounts of RhoA and Rac1.
So ELISA measures the overall concentration.
Yeah.
And flow cytometry gives you that single cell level data.
Exactly.
So what did they find in terms of overexpression after that mechanical strain?
Well, they confirmed a significant increase in both RhoA and Rac1 levels in the clinical samples.
After stretching.
Yeah, and this was consistent with what they saw in the cell lines.
Okay.
They also found that the baseline levels of RhoA
Yeah.
were already higher in the cancer samples compared to the non-cancer controls.
Interesting. So even without the stretching, there was a difference.
And there was this statistically significant correlation between RhoA expression and the stage of the cancer.
So higher levels in the more advanced stages?
Exactly. But they didn't find a correlation with age.
Huh. Interesting.
Yeah. And the paper gives specific numbers.
Oh, okay. Let's hear them.
For the changes after stretching.
Okay.
So in that stage 4 sample S2, RhoA levels increased from 0.5 picograms per milliliter to 1.55 after stretching.
Wow. A threefold increase.
Yeah, and in the stage 3 sample,
S3.
it went from 0.5 to 1.4.
Okay, still a significant jump.
Yeah, but the stage 2 samples showed much smaller increases.
Right.
Like in S1, it only went from 0.5 to 0.7.
Got it.
And the non-cancerous cells,
the FHCs.
Yeah, they showed a tiny increase from 0.5 to 0.6, which wasn't statistically significant.
So really highlighting that difference between the more aggressive cancers and the earlier stages.
Exactly.
And what about RAC1?
They saw similar trends for RAC1.
Okay, so also higher levels in the more advanced stages.
Yes, and a significant increase after stretching.
Got it.
In both the clinical samples and the LS513 cell line.
And did they include those p-values again?
Yes, they did. To show statistical significance.
And those p-values confirmed that the increases were significant.
Excellent. So they're being thorough.
They are.
Now I'm curious about those CM images.
Yes. The scanning electron microscopy.
What did those show?
They showed some really distinct changes depending on the stage of the cancer.
Okay, tell me more.
So that stage 2 sample S1, it appeared slightly flattened after stretching.
Okay.
And had increased surface roughness and more stress fibers.
So it was responding to the forces.
Yeah. But the stage 4 and 3 samples,
S2 and S3,
they showed more dramatic changes, more flattening, irregular shapes, and blebs on the surface.
Blebs, those are those bubble-like protrusions, right?
Yes.
Okay, and why are they significant?
Well, they often indicate changes in membrane tension
and
disruptions in the cytoskeleton, potentially from those mechanotransduction signals.
And they can be associated with early apoptosis?
So the fact that they were more common in the advanced stage samples suggests those cells might be responding differently.
To the stress?
Yeah, maybe experiencing more disruption.
And blebs have also been linked to increased motility and metastasis.
Right, so their presence is definitely noteworthy.
So all these changes.
The flattening the irregular shapes the blebs.
Yeah.
They seem to correlate with the stage of the cancer
Right.
and the mechanotransduction signals.
How did the researchers interpret that?
They suggested that as the cancer progresses,
Yeah.
the cells become more sensitive to those mechanical cues.
Okay.
So in the earlier stages, the changes might be more subtle, but in the advanced stages, those more dramatic changes could reflect bigger disruptions in the cytoskeleton.
Leading to increased motility and invasiveness.
Exactly.
And then finally, they use flow cytometry.
Yes, to look at those protein levels at the single cell level.
What did that reveal?
Well, it confirmed that most of the stretched cancer cells were expressing either RhoA or Rac1 or both.
Okay.
And